In 9.2, we're going to talk specifically about glycolysis and how chemical energy is harvested by the oxidation of glucose to pyruvate. So glucose loses electrons because it's oxidized. Glycolysis literally breaks down to the prefix glyco and lysis, splitting of sugar. And this is where we take a glucose molecule and we split it into two pyruvates. It occurs in the cytoplasm, and there are two major phases. There's an energy investment phase, where you have to consume energy to get the process going, and there's also an energy producing phase, where you get some energy out of glycolysis. Here we can see in this diagram that in your initial investment phase of glycolysis, you are in fact breaking down glucose into 2-pyruvate, and that's going to cost you 2 ATP. But in return for that, your energy payoff phase, you get 4 ATP. So as a result, the net ATP gain is 2 ATP. So 2 are used, 4 is formed, you wind up with 2 ATP as a result of glycolysis. This is substrate level phosphorylation. These next couple slides show a lot of detail in glycolysis, and it shows specifically on the molecular level what's changing to get us from the glucose molecule to 2-pyruvates. And for our purposes, we're going to focus on where ATP is made and what is needed for the process to start and what's produced, rather than the all the in-between steps, the intermediates that get us from glucose to pyruvate. So here in the first step is where we actually use one of our ATPs. So this is part of the energy investment phase. As we go from glucose 6-phosphate to fructose 6-phosphate, we are losing a carbon in our ring. Not much is happening in terms of ATP usage or payoff. In the next stage, we go from fructose 6-phosphate to fructose 1,6-bisphosphate, and here we are using our second ATP, and remember that only 2 ATP total are being used in the process of glycolysis. So now we should start to see ATP being made through substrate level phosphorylation. All right, this step is where we actually start to go from one molecule to 2 molecules. So at this point, your fructose 1,6-bisphosphate is going to be broken down by the enzyme aldolase into 2 molecules, 2 3-carbon molecules. And again, you don't need to know these details, you just need to know where is the ATP being used and where is it being made. At this point, we have electrons that are being lost by glyceraldehyde 3-phosphate. So 2 electrons become available. Those get picked up by our electron carrier, which is NAD+, along with 2 hydrogen ions so that the charge is neutral overall, to form 2 NADHs. Those are going to travel into the mitochondria, where they'll be used in the electron transport chain and the oxidative phosphorylation later on. All right, in our 7th step, we are getting our first ATP. So 2 ADPs are phosphorylized to create 2 ATP. Not much going on here. Again, we're seeing a change in our carbon structure. We are not concerned about what exactly is going on or what the names are. We're just concerned with what's being produced and what's being consumed throughout the reaction. And here is where water is produced, which is one of the products of cellular respiration. So in our 9th step, we do get 2 waters. And in this last step is where we get the last of our 2 ATPs. So we have 2 ADP that are phosphorylated to form 2 ATP. So this makes 4 total ATPs that are formed through glycolysis, and because we already invested 2, our net gain of ATP is 2 ATPs, and we end with 2 pyruvate molecules. So to recap, glycolysis happens in the cytosol, and if there's oxygen present, that's when your 2 pyruvates that were produced from one glucose molecule move into the mitochondria. However, before those pyruvates can be used in the next stage, which is the citric acid cycle, they need to be converted to acetyl-CoA, and that conversion from pyruvate to acetyl-CoA is what links glycolysis to the citric acid cycle. Here's your linkage reaction. So we have 2 pyruvates. Remember that 1 glucose yields 2 pyruvates. In the diagram, we're only looking at 1. But your pyruvate, as it crosses the membrane, more electrons are lost, so that pyruvate is oxidized, and NAD+ picks up those electrons along with the hydrogen ions and carries them to the next stage. And we lose a carbon dioxide group, add a coenzyme A to it, and so we wind up with the product acetyl-CoA, which can then enter the citric acid cycle. So this is the citric acid cycle in a nutshell. Just for your records, the citric acid cycle is also referred to as the Krebs cycle after the scientist Hans Krebs, who discovered it. So now that we have our pyruvate, which crossed the membrane, and when it crosses before it can go into the citric acid cycle, pyruvate will turn into an acetyl-CoA. And remember, we have two pyruvates, so therefore we have two acetyl-CoA molecules. So each turn of the citric acid cycle is going to refer to one acetyl-CoA. So in the end, because we have two acetyl-CoAs, we're going to wind up with double the products that we see in the cycle. So acetyl-CoA enters the citric acid cycle, and your acetyl-CoA gets oxidized, so we're losing electrons. Here we can see we have 3 NAD pluses pick up 6 electrons to form 3 NADHs. Those are going to travel to another part of the cycle of cell respiration, and they will be used in a different place in the electron transport chain. We produce an ATP as a result of this. We pick up more electrons. FAD is your electron carrier, picks up 2 electrons along with 2 hydrogens to form FADH2. Those again are going to travel to the last stage of cell respiration. When we get back to the beginning, we have our starting molecule. We can process the next acetyl-CoA. So one glucose molecule gives us 2 pyruvates, which gives us 2 acetyl-CoAs. So we wind up losing 4 CO2s, 6 NADHs are formed, 2 ATPs, and 2 FADH2s. Remember that we get 2 turns of the citric acid cycle because we have 2 acetyl-CoAs that are being processed. All right, in a little more detail than what the cycle is able to show us, your citric acid cycle has 8 steps. Each has a specific reaction with a specific intermediate that is formed. Your acetyl group of your acetyl-CoA is going to join the cycle by combining with something called oxaloacetate. This forms citrate, hence the term citric acid cycle. So the next 7 steps are going to decompose the citrate back to oxaloacetate so that you come back to your starting material that's needed to continue the Krebs cycle again. So whatever you end with has to be what starts the Krebs cycle so that you can replenish what is lost. NADH and FADH2 are your electron carriers. They're produced by the cycle, and their big function is to take electrons that are lost from the oxidation of acetyl-CoA and take them over to where they're used during oxidative phosphorylation, which is the 3rd step of cellular respiration. And this is just some beautiful details that you don't really need to know right now. Acetyl-CoA binds with oxaloacetate to form citrate. And as we go through, we can see where the electrons are lost, where the ATP is formed, and in the end, we have to wind up with oxaloacetate again as an end product because it is the first reactant in the citric acid cycle. So now we're starting to move into the last stage of aerobic respiration or cellular respiration, which occurs in the inner mitochondrial membrane. And this step is called oxidative phosphorylation. It's made up of 2 parts, the electron transport chain and chemiosmosis. So after glycolysis in the citric acid cycle, all of those electron carriers, NADH and FADH2, they are carrying electrons that are going to power machinery during oxidative phosphorylation that actually generate the ATP to do the work. That's what gives us all the energy that's extracted from food. The 2 electron carriers will donate electrons to the electron transport chain, which then powers the very last step, which is ATP synthesis, the creation of ATP through oxidative phosphorylation. The electron carriers take the electrons to the electron transport chain, which is in the cristae of the mitochondria, those inner folds of the mitochondria. They drop off the electrons into the electron transport chain, which is made up of proteins. The carriers, those proteins, alternate between reduced and oxidized states as they accept electrons and then donate electrons to the next step. So it's like this chain of proteins where the electrons are being passed. So one protein will lose an electron, the next will gain, and that's where the alternation of oxidation and reduction comes from. The electrons will drop in free energy as they go down the chain. So that means they have less capacity to do work as they move from one protein in the electron transport chain to the next. In the end, they're finally passed to oxygen, and hydrogen ions are picked up as well. And so the final product, or the final electron acceptor, is water, which is the other product formed in cell respiration. Here's an example of what the electron transport chain looks like. So you have a bunch of cytochrome complexes, and they have a bunch of well-named proteins that go into those. We're not concerned with what the names are. Just know that your electrons get dropped off by NADH. So now it's lost electrons, it's been oxidized. So NAD plus is going to go back to glycolysis. It's going to go back to the citric acid cycle to pick up and accept more electrons. The electrons travel down the chain. As they travel down the chain, they have the capacity to do work. And we'll look at specifically what work is being powered as a result of the electron transport chain. In the end, your final electron acceptor is water. Oxygen combined with a couple electrons and hydrogen ions forms water. This is slightly redundant, but follow me for a moment. So the electrons are transferred from NADH or FADH2, so they're dropped off to the electron transport chain. They pass through those proteins or cytochromes, each have an iron atom, and eventually the last acceptor of electrons is oxygen or water. The electron transport chain does not generate ATP. Okay, it does not generate ATP. That's what ATP synthase, the last step, does, chemiosmosis. The chain's function is to break the large free energy drop from the electrons getting dropped off to finally powering them to do work in chemiosmosis into small steps so that all the energy in glucose is not released at one time. If it were big ball flames, it would be an explosion. You'd have all the energy released at once. But when it's in small steps, it's more manageable, and we can actually harness that energy to do work. The very last step of oxidative phosphorylation and of aerobic cellular respiration is chemiosmosis. So your electron transfer happens, your electrons are dropped off into the electron transport chain, and this is powering pumps that are pumping hydrogen ions from the mitochondrial matrix into the intermembrane space. So what we're doing is we're pumping hydrogen ions against their concentration gradient. We're pumping them from a low concentration to a high one, so we're stacking them all on one side. The natural tendency for those hydrogen ions is to move back across the membrane, to diffuse back down their concentration gradient. As they move through, there is one specific channel that they move through to get to the other side of the membrane to go back to equilibrium, and that is the ATP synthase, which is an enzyme. ATP synthase uses the exergonic flow of hydrogen ions, okay, so no energy required, it's spontaneous, to drive the phosphorylation of ATP. So this takes an ADP and a phosphate and sticks them together into ATP. This is, in a nutshell, chemiosmosis. And the use of energy in a hydrogen ion gradient or proton gradient is used to drive cellular work. I think this is much more pretty in picture form. So let's talk through the same exact thing we talked about, but let's use a diagram to explain. So in your electron transport chain is driving the movement of hydrogen ions against the concentration gradient. So it's moving it from the mitochondrial matrix to the inner membrane space. So there's not many hydrogen ions on this side of the cell, and there's a lot on the inner membrane space. So what's going to happen is, to go back to equilibrium, these hydrogen ions are going to want to go back across the membrane. So they're going to want to move back across the membrane. In order to get across the membrane, they're going to go through something called ATP synthase, which is this gigantic motor-looking thing. As they diffuse back across the membrane, they travel through ATP synthase. The hydrogen ions enter the ATP synthase, and it causes the ATP synthase to turn. Each turn of the ATP synthase takes one ADP and one inorganic phosphate, and it sticks them together to form ATP. So the electron transport chain drives the hydrogen ions against their concentration gradient, and they then diffuse back through the ATP synthase to take ADP and P and reform ATP. So pumping hydrogen ions across the membrane using the electron transport chain as your driving force couples your redox reactions from electron transport to ATP synthase. As we pump against our concentration gradient, we are creating the capacity to do work. We're creating potential energy. So now it takes no energy whatsoever for those hydrogen ions to come back across the membrane and back down into equilibrium. This is a spontaneous reaction, and because you front-loaded it with the electron transport chain, you've created what's called a proton motive force. So those protons, as they travel back through the ATP synthase, now have the capacity to do work. They are powering the phosphorylation of ADP to form ATP. Let's look at a pretty picture to try to describe what the heck I'm talking about. So this portion right here, these proteins, are part of the electron transport chain. So NADH comes by, drops off his electrons, and electrons are going to travel through the electron transport chain. FADH2 drops them off as well. So you have these electron carriers dropping off the electrons. The electrons have a lot of capacity to do work, and the energy that they have is driving these pumps that are pumping hydrogen ions to one side of the membrane. So we are pumping against the concentration gradient. It requires energy. Electrons provide the energy to do that. So what we're doing is we're taking all of our hydrogen ions and stacking them on one side of the gradient. Well, their natural tendency is to move back across the membrane. And to get back across the membrane and back at equilibrium, they're going to travel through ATP synthase. As the hydrogen ions travel through ATP synthase, you get a turn of the machinery, and your ADP and your inorganic phosphate come together to form ATP. And this is how we get most of the energy in our cells to do work through cellular respiration. All right, so your energy flow starts with glucose or any organic compound. The electrons that are lost by the breakdown of glucose are picked up by your electron carrier NADH. They're dropped off at the electron transport chain, which creates your proton motive force, all those hydrogen ions on one side of the membrane. And then finally, it is used to create an ATP molecule. So you have that energy stored in the ATP molecule. 40% of the energy that's available in glucose is transferred to ATP during cellular respiration. So it's 40% efficient, which is really low when you think about 100% efficiency possibility, but considering it's a machine, it's very, very, very efficient. So what you wind up with in the end is about 38 ATP total, and that's a result of aerobic respiration or having oxygen present. So breaking down glucose into 2-pyruvate, 2-acetyl-CoA, goes through the citric acid cycle, and eventually your last step, oxidative phosphorylation, which has the ETC, electron transport chain, and chemiosmosis. These are your main steps. What you really need to pay close attention to is the capacity to do work. So you're releasing these electrons into NADH and FADH2. They are carried over to the electron transport chain. The whole function is to release these electrons so that you can harness them to do work in the electron transport chain, so that you can create a proton motive force, so that you can drive the synthesis of ATP. Remember that glycolysis has a net ATP gain of 2. Citric acid cycle makes a couple ATP, so those two steps together make 4. But it's that last step, oxidative phosphorylation, that provides 32 to 34 ATP. So most of your ATP is coming during that last stage with the electron transport chain and ATP synthase. So overall, you're making roughly 36 to 38 ATP, give or take.